Hi, thank you very much. I'm Masha. I hope you can hear me well. And I think I'm going to add even more complexity to what happens to drugs after they are launched. That is really related to their taste. So drugs will only work if we actually take them. And in fact, I'm going to share with you one example. This is an example of anti-HIV drug. There are almost 2 million children under the age of 15 living with HIV, and only 50% of these children take the antiretroviral treatment compared to much higher percentage of adults living with HIV. And this is due to a very aversive taste that these drugs have. And actually, it led Gilead Sciences to collaborate to design specific pediatric formulations to eliminate bitterness of these two drugs. So this is not the only example. In fact, since 2018, the FDA expects that all medicines that have the potential to be given to children should be assessed for their palatability, so how tasty or not aversive they are. And this is similarly also for the European Medicine Agency. So it is well known through our human history that many drugs are bitter. You even can call it the bitter potion, or if it's not bitter, it will not cure you, and so on. And the typical process in pharma today is that you go through a very convoluted, very sophisticated drug discovery process, from the screening, virtual screening, cellular screening, then preclinical phase with animal models, and then clinical phases. And actually, currently, only in these clinical phases, or even post-marketing, becomes or rises the problem that the drug actually might be intensely bitter, and then you have to go back to reformulation and to try and mask the aversive taste. And so we wanted to ask whether we might be able to do better. Maybe we can change this process and to be more efficient and to foresee the problem with the taste early in the process. So how did we do that? So for several years, we've been working on bitter taste, and we have established a database of bitter molecules that we call the bitter DB. And this is publicly available. You can search it. And in fact, there are many users that use this database, both in food industry and in pharma industry. And currently, it has information about about 1,000 of bitter molecules. And we will be updating, releasing new version of this database soon, which will have almost double number of molecules. So once we know what is bitter, we can start developing predictive tools to also to predict what else might be bitter. And so we did this in work a few years ago, where we developed a machine learning tool that we called Bitter Predict for predicting taste from chemical structure. But actually, that might not be sufficient, because we may not only need to know what is bitter, but we actually need to know what is intensely bitter. We really need to look for those molecules that are very bitter, because otherwise it's not a problem. So then, in order to develop the bitter intense algorithm, we had to gather additional information. So data, of course, is key when developing database classifiers or predictors. We really rely a lot on the data that we have. And so here we partnered with companies that had proprietary data, and we had additional data that enabled us to divide our data sets to very bitter molecules and not very bitter molecules. And then we were, and just to show you an example, there are difficult chemotypes and difficult chemical structures that can contribute to bitter taste, but we noticed that not very bitter families of molecules of compounds can be very diverse. And in the example that, or at least in the data set that we had so far, the triterpene saponins were particularly enriched in the extremely bitter subset. So once we had these bitter and not very bitter and not very bitter molecules, we could use this data for training a machine learning algorithm. We calculated several descriptors for these molecules. And here we used XGBoost, extreme gradient boosting algorithm, that learns from the mistakes in previous iterations and also is known to work well with small and unbalanced data sets, which is what we had. And so this bitter intense method reached the accuracy which was over 80%. Using in holdout set that we had. And so these were quite satisfactory results, and which enable us now at this stage to predict the bitterness of a molecule in very early stage. So it even doesn't have to be synthesized, because basically even in virtual screening, we can already flag the molecules that have the potential to have intensely bitter taste, and then the whole process, including the preclinical phase and clinical phases, can take this into consideration and already consider formulating to reduce the bitterness. So it can already work with the formulations and be aware of the problem very early. But this is probably not enough, because we actually want to be able to understand even better what is responsible for the bitterness of the molecules. And maybe if we want to mask bitterness, it would also be good to understand which are the receptors actually that are responsible for the bitter taste. And in fact, the receptors for bitter taste are called TAS2Rs, and they belong to a family of GPCRs, G protein coupled receptors. In fact, also sweet and umami taste receptors are GPCRs, and also olfactory receptors are GPCRs. But I'm going to focus here on the taste, on the bitter taste receptors, these T2Rs or TAS2Rs GPCRs. And what is very interesting about these receptors is that it's a quite large family. There are 25 different bitter taste receptors in human. And another interesting point is that there is huge evolutionary diversity. So other animals, other organisms have different number of subtypes of these taste receptors. So what we would like to be able to do is not only to predict the bitterness of a molecule or intense bitterness of a molecule, we actually want to know which receptors are responsible for that bitterness. And as I said before, humans have 25 different bitter taste receptor subtypes, and one ligand can activate many bitter taste receptors. One receptor can have many ligands, and we want to be able to assign our molecule to all the bitter taste receptors that are relevant for it. So how can we do this kind of matchmaking? So what we decided to do was take an example or be inspired by something that is often done in recommendation systems. So often we, when buying products, for example, or when trying to match a client to a product or music to music listener, there were systems that were developed, a recommendation system developed for that, and we took some ideas for these kinds of approaches. So in our case, we can consider something like a matrix where we have different ligands and different receptors. And for some of these pairs, we have information that was measured experimentally, and either if it was found as a match or a molecule activating the receptor, or it was found as not activating, shown in red. And then we have these empty slots. So in some cases, there was no measurement, and this is what we want to be able to predict. And in even more difficult scenario, we want to have a molecule that was never measured with any of the taste receptors, and we want to be able to assign that molecule to the bitter taste receptors. So the information we have is this experimental information about what was already measured, and we have developed neighbor-informed chemical features. So basically what we are going now to measure is to measure different kinds of similarities using chemical similarities or sequence similarities for receptors. And we are going to weight these similarities based in the following way. So for example, if we have an incoming molecule, and we want to know if it would be able to activate a bitter taste receptor. For that bitter taste receptor, we already have experimental data for many different other molecules. Some activated that receptor and some did not. Now we're going to measure the chemical similarity of our new ligand to those molecules that activated the receptor, and we're going to record the highest similarity to activating ligand, and then we're also going to record the high similarity to non-activating ligand, and in addition, we're going to have the sum of activating ligand similarities and sum of non-activating ligand similarities. So here it's a combination of chemical similarity, but it is weighted, or it has the information about what already was shown to interact with that receptor or already shown not to interact with that receptor. And then in addition, we have ligand properties such as charge, bond count, ring count, and so on, and also have receptor properties such as sequence-based and structure-based properties. For example, information about the cavity, the binding site, site volume, charge, surface area, and so on. And here again, now we're going to use a tree-based model with boosting to predict for any ligand all the possible targets, all the predicted targets within this family. And I gave a title of progress and challenges, so I'm going to mention the challenges here, which include, for example, that we have a sparse data set. So actually we have few known interactions and a lot of holes in this matrix, and unbalanced data, so many more true positives, true negatives than true positives. And we also have very low resolution of the structures of the receptors. And also we don't have binding assays in this field, they're only functional assays, so activation or non-activation, we don't know about binding per se. But nevertheless, here I'm showing precision as a function of recall and also average precision. And you can see that we were able in the bitter match to we were able to reach average precision of almost 80%. And this is very, very good, both considering the challenges, but also when comparing to a naive approach in which I would say, okay, let's say that the receptor that is activated by the largest number of molecules known so far would be my first guess. So this kind of naive approach is only giving us an average precision of 50%. And so the method we have developed, taking into account all the properties and known the known experimental information, is doing much better. That was not enough for us because the results, so the performance I showed you before is based on a set that we left aside as a test set, but we actually want to be able to prospectively predict something that was never measured, not known, not to us, not to anybody else, make the prediction, and then do the experiment and see whether indeed we are able to generalize and to be able to predict for new molecules their interactions with bitter taste receptors. And this is what we did here in this work. 12 compounds, we predicted the interactions with more than 20 human bitter taste receptors, and these were tested in functional assays experimentally. And you can see that we had these are true positives, 16 true positives, over 200 true negatives, and a little bit of false positives and false negatives, resulting in precision of 80% and recall of over 40%. So this is very good, and we also wanted to know what is the secret of success here. And what we found is that the most important features that contribute most to the good performance of our predictor are those chemical similarity features that I described before. So chemical similarity to ligands that do not activate the receptor in question, and chemical similarities to ligands that do activate the receptor in question are really important for the success. And then come the identity and similarity of binding sites of the receptors, and then some features of the receptors as themselves. So when we actually wanted to look at which are the taste receptors that are activated by most of the ligands, both known to be activated or now predicted that we predict to be activated, and in particular pharmaceutical drugs from drug bank, we did this analysis, this prediction, and we found that T2R14 or TAS2R14, so this receptor, one of the 25 human bitter taste receptors, there's this particular one that really is activated by many pharmaceutical drugs, or at least we predict so, and also known to be activated by many compounds. And that's what gave us the need and the will to see whether we can inhibit this taste receptor. And also we would be very interested in understanding what is the physiological relevance of this receptor. So first, the discovery of an inhibitor. We here worked on a kind of iterative approach where we started with a set of known agonists and very few antagonists that were known for this receptor. And then we used induced feed docking to generate different conformations of this receptor, starting from a model, not an experimental structure, a homology model, and then docking into the different models of this receptor, known ligands and decoys. And then we could rank, or we could choose the best. So we have the induced feed docking, and then we can choose the models that are doing the best job in differentiating between true positives and true negatives. And so then choosing this winning model, we can then go on and use it for virtual screening, structure-based virtual screening of database of compounds. And here we used enamine. At that point, there were 20 million compounds that we could virtually screen against the best model we had so far, and then select a few molecules for experimental testing. This is what we did in addition to also medicinal chemistry efforts at the lab of our collaborators, and all together. And then, of course, each time we update the set and we repeat this process iteratively. And what we were able to obtain after a couple of rounds like that is we were able to find 10 antagonists and 130 new agonists of T2R14, and we could also use bitter match to predict that actually they are selective for T2R14 in particular. So what is nice here is that now we screened the 20 million libraries. We purchased actually only 30 compounds, and 3 of them were performed as confirmed experimentally as antagonists, and there were also some additional agonists. But we really were particularly interested in antagonists because we want to be able to inhibit this receptor. And in fact, these antagonists were already used by other researchers in other labs. So this is already being used as a chemical probe for inhibiting the activity of this receptor. Why is it so interesting? So in addition to everything I told you, it's also so interesting to work on taste receptors because they are not only expressed on the tongue, they are actually also expressed in different tissues extraorally. And what they are doing in the other tissues is not completely known. We know, well, if I have time during the question and answer, I will talk about this a little bit, but it is definitely very interesting to understand what they are doing, and therefore having an antagonist that is able to inhibit a particular taste receptor is very useful in order to explore the physiological role of these receptors, and also maybe help us perhaps understand polypharmacology or off-target roles of bitter drugs that work through their main target, but probably also through the bitter taste because they're bitter. So maybe also through bitter taste receptors. So what I showed so far is that we have a kind of bitterness masking pipeline starting from structures. We can predict intense bitterness, and then we can bitter match to really understand through which of the 25 different bitter taste receptors this bitterness is achieved. And then after confirming in cell-based assays that we were right about the bitter taste receptors, we can then also use structure-based approaches to discover antagonists for the particular bitter taste receptor that's interesting for us. So again, progress and challenges. So we have 25 different taste receptors in human, as I said, and here I'm showing, and mice have 30 different bitter taste receptors. Here I'm showing the precision and recall of bitter match, but now shown individually for different taste receptors. And you can see that there are many taste receptors, bitter taste receptors, for which we have both good precision and good recall. But we also have others for which we don't have such good results. So there's still work to do, and we're working on this. And another challenge that I wanted to mention is that even though we have bitter taste receptors in human and bitter taste receptors in mice, in fact, they're not so similar. And when we look at the mouse analog or supposed to be ortholog of the human bitter taste receptor TAS2R14, we see that only a small portion of the ligands is actually common to both of them. So this is challenging because usually we use mice as models for humans, and here we know that the model is probably not so great because we have such big differences, and this is definitely a challenge, and this is why we're also working in parallel both on human and on mice and other species receptors to understand it better. So I will summarize. I presented the bitter DB, a bitter database of bitter molecules and receptors that we are maintaining and are expanding, and we welcome everybody to use it and also contribute information if you know information about bitterness that is not there yet. I presented very briefly bitter predict, but mostly I talked about bitter intense. So a machine learning tool to predict intense bitterness of molecules based on their structures. And then the bitter match. So it's matching ligands to receptors by using information about ligand properties, receptor properties, and also similarities between ligands that were already shown to activate or not to activate receptors. I also mentioned the recent work that we did to find the bitter taste receptor antagonist, and these can be used both for masking bitterness and for studying the roles of extraoral taste receptors. And one kind of take-home message is that we made a lot of use of data. So experimental data about ligand receptor associations, it helped us develop these neighbor-informed features that turned out to be crucial for bitter match performance. We used it also for the refinement, for induced feed docking and refinement of the different three-dimensional models of the bitter taste receptors. And we also then used it again for evaluating which of the possible models are best for differentiating or giving different ranks to known ligands and true negatives. And so really, I think this integration of experimental and computational data is extremely important. And I would like to thank you for giving me this opportunity, and the funders and the team of my students, and also to mention that we'll have openings soon if anybody of the young audience participants is interested. And thank you very much.
Thank you, Masha. We have about 3 minutes, and I see now one question and then two another questions. First one, how does the perception of bitterness vary between different peoples?
Okay, so there is a variation. One very well-known example is that there is a genetic difference in T2R38 receptor, one of the receptors, there is a taster version and non-taster version in that particular receptor. So if you have the two copies from both of your parents, non-taster and non-taster, you will not be able to feel bitterness of a particular type of bitter molecules, but still you will be feeling bitterness of other molecules because you have the other receptors still functioning as usual. In other cases, genetic variation can affect, but not like this, not like you're complete non-taster. And then there are additional effects of age has an influence on how strongly you feel. And there are also cultural differences. So you might be able to feel the bitterness, but you don't treat it as something as bad as somebody else. And that on this value that is given to bitterness, there is a lot of impact from the cultural and your experience in life, let's say, and what you're used to. But in terms of just saying if it's bitter or not, it is kind of consensus. Most of the people would say about the same things, that they are bitter.
Thank you very much. Another question. So there is some, dear Masha, your work and presentation is great. Said Azimi. And
thank you very much. Thank you.
The question is, next question is from Rustam Taktashev. Do you use F1 score or MCC to evaluate the quality of predictions of your model? The set seems to be imbalanced since there are a lot of true negatives.
Yeah, absolutely. I didn't show here, but yes, we used balance accuracy and F1. Yeah, because exactly because of what you said, because we have unbalanced data sets. Yeah.
Thank you very much. So I did not see any questions. So thank you very much.
I have also a question. I'm not a specialist, but from taking this opportunity to also to run this section. Masha, please explain us if speaking about some drugs which are bitter, can you modify these drugs to make them like not bitter, not changing their functional properties?
Okay, so what typically people do is coating, right? So you coat it in a tablet, and then you don't need to feel in a capsule, and then you don't need to feel the taste, and then you don't change the chemistry inside. You're just doing the coating, but it's a problem for little children and for old people if you cannot swallow a big pill. So there is a solution for many cases, just the coating solution, but it's not working for kids. Another thing that people are often using is liquid, like syrups, where a lot of sugar and the flavor is added, and it also helps in some of the cases, but still not always. So it's still in some cases you have a situation where kids do not want it. And by the way, also animals. Sometimes you also have the same problem with veterinary drugs. Now, I don't, I think it's a very good question. I don't, well, we have to talk about it later.